So thanks for everyone to coming to my webinar. It's really honored to present my recent publication with the break. I'm Yura, as she introduced. I'm working at the Free University of Brussels in French-speaking area of Belgium, and I'm currently working as a bioinformatician with Cedric Blancpain. So the topic, the title of today's presentation is a collagen signaling and matrix stiffness regulate multipotency in glandular epithelia stem cells in mice. And mainly we will focus on about how the mechanical stress is affecting to the cell fate changes in the mammary gland and prostate. Before we are starting, I will try to introduce a little bit about the mammary gland and prostate biology. So as you could see here, actually the mammary gland between human and mouse are quite similar. They're composed of ducts and connected to nipple, and then there are terminal ductal lobar unit on the end, and there is surrounded by stroma. And if you're colored as a horizontal, we can see that this kind of a structure, like basal cells are all the outside and they maintain the shape. And inside there is a columinal cells. So those are classified as the two types, which is sensitive to estrogen signaling. We usually say ER plus, and which is not sensitive at all on the ER signaling, which we say ER minus. And when we cut this vertically, you could see that the structure like this, the adipocyte, the stroma are around, and there's a basal cells, which is as a pink, and luminal cells, which is in the very inside and marked as a purple. However, when we move to the prostate of the mouse is composed of different part, which is nearby the bladder. As you can see, there is called ventral, lateral, dorsal, anteral, and those are also surrounded by the other part, like the seminal, vesicle, and ductal defense. And still, it has a similar structure as a mammary gland, but it doesn't have any ER plus, ER minus luminal cells anymore, since it's not related to estrogen signaling at all. So it just have a basal cells outside and luminal cells inside, and the basal cell we usually define with the K14, K5 keratin gene expression, and luminal cells with the K8 gene expression. And when we try to see as the images, which is called it the vertically, it look like this, and horizontally, it will look like this. And one thing I have to point out is that compared to the mammary gland and prostate, there's a structure between adult human, adult mouse are totally different. As you could see that even how they define the part of the prostates are different. However, when you see that the inside of how the cells are composed, still they are same, like basal lamina outside, lumen inside there, and then the inside of lumen there is halo. So it looked like a cube, and then there's a secretions or some other materials can move. So actually, this is the kind of like comparison how they can be ortholog between human and mouse, but it's not exactly same. So since I was talking about the multipotency, maybe we have to understand how the cells are multipotent and becoming unipotent. So in a mammary gland and prostate both, when it is an embryo and it's a fetal developmental status, it's a multipotent. So there is no basal and luminal clear definition, to be honest, because it can be both. However, on the mammary gland right after birth and the prostate in the basal mouse, after 21st day of birth, which we call puberty, those cells are becoming unipotent. Therefore, the basal cells only can make basal cells, and luminal cells only can make the luminal cells. So in our lab, so even like before I started working in our lab and during the work in my lab, so we tried to see how this unipotent cell, the adult cells, are getting the remultipotency, let's say. And the very first experiment they tried was which we call transplantation. So what they did was taking the only basal cells from mouse mammary gland and put it in a clear fat pad. And we observed that this condition, the transplantation of the only basal cells can make the luminal cells that even form the full structure of mammary gland. However, which we only put the luminal cells or basal luminal cells together, it didn't make any differences. It just kept unipotent. And afterwards, that the same authors are doing the experiment because PIK3CA is the one of the famous oncogene, which is really commonly mutated in breast cancer. And what we saw was that if there is a PIK3CA mutation in the mammary gland cells, this adult basal cells adult basal cells can have a multipotency. However, the direction is luminal to basal cells. Therefore, from this idea, what we tried in 2020 was we chemically ablated luminal cells. So it means that we kill the luminal cells selectively. And these adult basal cells are started moving to make the luminal cells. And furthermore, the one of the thing we find, I already tell that in the embryonic status, those cells are unipotent. And what we found is that not only the multipotent cells, we also find the hybrid status between basal and luminal cells, which express both markers of basal and luminal. So we try to understand how this multipotency mammary gland and prostate, let's say, they are defined, how they can be reactivated, and how they affect in the system of the mouse. The one thing I have to tell before we are really moving to the result of the paper, actually after birth, these unipotent cells in the mammary gland is becoming basal and luminal first. And these luminal stem cells still go further to the ER minus and ER plus cells based on whether they are sensitive to estrogen signaling. Usually this stuff happens in puberty. So this one is already happening after birth, like they are separate to the basal luminal. And after it goes to the puberty, it starts to be clear whether it will be sensitive to ER signaling or not. So I will start to move to the result of the paper. So from here, I will talking about the paper which we published in like around a month and a half ago. So as I already explained to you on the previous slide, we already knew that the different conditions of which can reinduce adult multipotency in mammary gland. And then the one of the experiment we did on the prostate is that because we already know compared to duct, tip is more multipotent. So what we did was taking the 13 days after birth of the mouse and then take the basal cells only and try to see what kind of genes are upregulated in the tip compared to duct to define the multipotency signatures. So when we are comparing all these 5 different conditions which is related to adult basal multipotency, we found that there are 3 genes commonly upregulated in all these 5 conditions, which are the collagen 1 and collagen 3 genes. And furthermore, when we try to see that the gene expression in this EMP data, which is here the embryonic progenitor, we could see that these collagen genes are specifically expressed in this hybrid condition, as well as when there is a basal cell transplanted and when there is oncogene activated, lumina ablated, and even in the tip compared to duct of the prostate. So we confirm that these collagen genes might be related to the multipotency regardless of different conditions. And this data generated by 2 other first authors of this paper. And then these people are doing was that since we already know the collagen will be the one of the factor controlling multipotency. So what they tried to do at the beginning was taking the mammary gland and making the organoid and put it in a gel with collagen, which is 4 milligram per milliliter. And in the matricial, we saw that the kind of clear definition of the cells, since the tomato is chasing so if I explain a little bit more, here the tomato is chasing the luminal cells from basal cells, and K8 is the pure luminal. So if there is a co-expression of those 2 markers, it means that these luminal cells are from basal cells, which is derived from adult multipotency. So in the matricial, we didn't see any kind of a signal which is going to basal to luminal. However, in the collagen cells, as we start to see the co-expression of the tomato and K8, which indicate these luminals are arise because of multipotency. And if we are using the different concentration of the collagen gels, the collagen for the organoid gel, we see that there is a kind of significant increase of the basal derived luminal cells, as well as we do the same experiment on the prostate. We exactly see the same result, and same significancy. So here, what we get the idea was that from the first result that maybe if we're adding some more stiffness on the organoid culture gel, whether we could see the same result or not. So here the mammary gland again, we try to use a PEC, which is kind of composed of also composed of the laminin too. And when we put this in the organoid gel, we could see that the kind of stiff gel, which is like giving the pressure to the cells. And we also see again then there is no changes between 2.5 and 3% of the concentration, but when we increase the 7%, we see that there is a significant increase of this beta-derived luminal cells. And as well, when we are doing the same experiment on the prostate, we could see the same tendency, and it's already started getting more significant from the 4%. So here, what we decided to do was a single cell sequencing to understand how this different stiffness on the organoid are making the multipotency, and whether these multipotencies are composed of the different intermediate. So here we only did the mammary gland because the prostate we failed experiment. It's really hard to get the cells, the enough number of cells for the single cell. So here we define the 4 different conditions, which called matrigel, which we use as a control, soft collagen 1 and stiff gene. And we try to do the cell hashing to pull matrigel collagen as a one set, soft and stiffness one set. So for the cell hashing, if I briefly speaking, that we are pulling the different samples in the one tube and try to do the round of the sequencing in the one lane. But here, rather than using the barcode only, we use a kind of antibody, which one has a feature barcode and then the sequencing barcode. So after we do the sequencing, we know which sample is from which barcode. So the first thing we did was individual analysis, and from the matrigel, soft collagen, and stiffness, the clear difference is that already in the stiffness, we find several different hybrid status, which is called basal luminal, and a hybrid of the ER plus and ER minus cells. And also, I mean, we still see that the different hybrid from the matrigel to stiff, and the clear differences that from the increase of the stiffness, we start to see more hybrid. And also when we are defining the hybrid by the expression of the basal cell marker and luminal cell marker, since hybrid is the marker, the hybrid cells are expressing both markers, we see that the number of cells are getting more by the stiffness. And when we quantify this hybrid, which is higher than 65% of expression for both markers, we see that it's like 3 times higher compared to the control condition. So we could say that the stiffness can induce the different hybrid cells in the organoid of mammary gland. So we decided to integrate this data because that we would like to compare the gene expression, and we wanted the further analysis, such as like a pathway. So when we integrate all for this data set, the one of the interesting is that we find one cell called basal prime luminal, which can go both direction of the lumina ER minus, lumina ER plus. And then when we use a slingshot, which is used for inference of lineage trajectory in the single cell, so try to understand how the differentiation path can be defined, we find that it's always starting from basal and then going to the prime and directly jump to ER plus, or passing by hybrid basal ER minus and going to ER minus and stop it, or it can go further to until the end, which is lumina ER plus. So we understand that maybe then there is a jumping to directly either to ER plus or going to ER minus or going to full path. We try to understand how this hybrid status can be defined. So what we tried to do was the kind of comparison between the basal versus hybrid basal ER minus, which is on this cell, like which is in the middle of the way, and compared to basal. And also we do the same thing on the collagen condition, stiff condition, and then we see that in the basal, definitely we see the bunch of basal cell marker like keratin 5, ACTAC2, the myosin, for example, but still we see the laminin collagen which are related to this gel. And when in the hybrid conditions, we see the kind of typical marker of luminal signature like EpCAM, K8 keratin 18, but we also see that the L5, which is the master regulator of the lumina ER minus cell fate, and as well as a cludin, which is still related to the basal cell. So it means that the hybrids are going to already luminal, but still has a basal status. And the next question was on how about these 3 collagen genes when we saw at the very beginning, because we didn't find those 3 genes in this both DG set. And then what we find is that these 3 collagen genes, which we define as a multipotent signal, those are really exclusively upregulated in the priming condition, which can jump to both different the different luminal population. And meanwhile, we were doing this paper. One of the editor was and one of the reviewer was asking, is there anything common with the previous study? So we quickly take the marker genes, which are like exclusively upregulated on the condition of the prime cell and hybrid of basal ER minus. And we actually saw was that there is not so much of overlapping with the condition, which is a ablation of luminal or embryonic progenitor. And even though we do with the other conditions, we didn't find any genes, and those genes are still the collagen genes, which we already know before. And there is one of the groups studying about the luminal primed basal cells. It means that the luminal can make the basal cells without any intermediate. And they also did a single cell. So what we did was try to take their signature of a luminal prime basal and see whether there is enrichment. We didn't find any enrichment. So by this analysis, we can say that our hybrid is more unique upon the mechanical stress. So here then the next question is after, okay, we understand what kind of genes are upregulated and what kind of things are common or not common between the previous studies. And the next question of us is that then what kind of transcript factor is controlling this whole downstream genes? So we tried to see what kind of genes in these basal cells are upregulated in the collagen condition, stiff condition compared to control. And commonly we find the gene FOSB, those marked the transcript factors are AP1 transcript factor. So that we were started thinking, maybe then the one of the important factor will be AP1, but the problem is AP1 is also composed. This AP1 should be composed of different the axis. So we decided to how this the regulator of the axis is composed of. So we start with AP1. So after we try to understand what kind of genes are affected by this multipotency, so rather than we do this simple way of a DEG, we decided to make the kind of linear model. Since we have a different stiffness, which is increasing. So, and also definitely there will be the effect of the collagen itself, since we use a collagen for the collagen gel. So we try to make this linear model and try to find, are there any genes whose expression is gradually increased or gradually decreased by stiffness? But in this model, the limitation is that it's really hard to apply the cell identity because the factor information was not really well accepted, and even though we put as a covariate. So we decided to run this model separately on the 3 different conditions, which is showing the strong basal multipotency. So this is the result of the modeling, and then we could see that there are definitely the genes which you already saw on the DG analysis of collagen gel and stiffness condition. So here we confirm that the AP1 gene expression and the activation of the AP1 is promoting this multipotency, and maybe they will be also working in transcription further. And the one thing I have to point out here is that I marked like several different ribosomal genes, including UBA52, which is actually known as which is the sign of regulation when there is a translation of the stem cells. So that's also the one of the findings that we can say definitely our samples, our data sets are related to the multipotency and then the regulation of the stem cells, as we say on the beginning. Definitely in this model, as explained, there is AP1 and also as well as known as the factor for the stem cell translation regulation. We also found the several transfer factors, including KLF6 and KLF2, so which are changing their gene expression by the stiffness, but it's not like a gradual. It's more like jumping from the collagen and stiffness condition compared to the other 2 low stiffness condition. And since we only have the single RNA-seq data, so if you'd like to know what kind of transfer factors are activated, we can use the tool called Cynic. to do the prediction of the motif which are activated in our data. And I just briefly speaking about their notion. So they are trying to find which genes are co-expressed with the transfer factor. Like they will try to find the same patterns, like upregulation of transfer factor, upregulation of their target genes, and vice versa. And whenever they define these transfer factor groups, they will move to the database they curated, and they will try to see whether this gene has a binding, these genes are having a binding site of those transfer factors, or whether they are near to the promoter or not. And then they will define which network we should thinking about. Afterwards, we can put it as a AUC value, which is a score area under the curve. So we use these tools to define what kind of transfer factors are activated. This is a whole map. And then definitely we start from basal, and then those transfer factors are already well known in basal cells in the mammary gland biology. And then we start to see that already the Jun and then KLF2, which we already saw before, CREP5, which is a sulfate related transfer factor. Those are started activated in the prime population. And if it goes directly to luminal, those two are also well-known luminal ER plus transfer factor. And when we go to the hybrid of the basal ER minus, which go to the ER minus in the end, actually those two factors are again related to stem cell regulation. And they show the ER minus, that those two are also the cell specific. And again, we see on this hybrid ER plus to ER minus, we see the transcript factor we define on the cynic and on previous DG analysis again. So we can say the AP1 as well as the other stemness regulating factors are important in our data. And when we are checking the regular activity, there is an example as AP1 families, and we could see that there are differences between mature soft unipotent condition, collagen and stiffness, which are multipotent condition, and it's a significant. And then one of the reasons we moved to the integrin beta 1 is that actually when we have the multipotency in mammary gland and prostate, those kind of glandular epithelia, it's already well known alpha 2 beta 1 integrin is activated. So we were starting with blocking the beta 1 integrin by antibody. And then we could see that when we treat this blocking antibody, there is a huge decrease, like significant decrease of basal-derived luminal cells compared to mature soft compared to these two light stiffness. We also see the same condition on the prostate. Then afterward, whether one of the review, because we only showed this part, and then one of the reviewer was asking, is it the FAC signaling important to control this multipotency? So we use the FAC inhibitor, which is a PF 573228. And as well as we decided to use the alpha 2 beta 1 inhibitor to be clear whether this is really alpha 2 beta 1 or the integrin beta 1. And then those two inhibitors, the pharmacological inhibitors, we again could see that there is a significant decrease on the multipotency when collagen stiffness condition, and as well as the prostate. And also when we treat the FAC inhibitor, usually we expect that the mammary gland or prostate will look like this, and there is a multipotency, and even though there's multiple, they will make the kind of new branch like on the upper. However, when we treat a FAC inhibitor, we don't see any kind of morphology anymore. It means that there is no multipotency which can make the full structure. And as well, yeah, here this is the kind of like well-known shape of the mammary gland and prostate. But when we treat the FAC inhibitor, the organoid usually don't make the feature, and they will just aggregate as a one cell. So we could see that this is kind of clear signal that the FAC signaling is also important, and it's composing the one of the axis for the multipotency controlling. Then definitely, since we said that the Jun and Fos are the one of the signal, which is AP1 family, they are important for the which is important for the multipotency. So the other question was that when we treat those kind of inhibitors, is there any decrease of those gene expression? And then I would rather say yes, because here you could say that the Jun and Fos, which we only did on the mammary gland though, So we could see that there is a quite a clear decrease of the gene expression of the gene and FOS after treating the FAC and alpha to beta 1 inhibitor. And if you're using, I mean, even though there is a less expression, sometimes the transcription factor can work. So what we tried to do was that we really inhibit the AP1 function by using a pharmacological inhibitor. And then we again could see that there is huge decrease of the multipotency of collagen 1 and stiffness. And these are all saying that not only integrin beta 1 and FAC signaling, and then AP1. So this axis is controlling really the multipotency from the stiffness on the mammary gland and prostate. So if I summarize my result into the one page, so we say we take the mammary gland and prostate making the organoids, and we are applying the different stiffness, let's say mechanical stress, and then we see that by the increase of the stiffness, we see that integrin beta 1, FAC, and AP1 activation. And then we also see the multipotency. So where there is more multipotency when we apply the higher stiffness. And the differentiation path is starting from basal and going to prime the luminal cells. Either it directly jump to luminal ER plus, or passing by the hybrid and going to ER minus, or it can be stopped, or it can go further to or reach to the luminal cells or ER sensitive luminal cells. So thanks for the talk today, and I need to acknowledge some of my colleagues, like my supervisor, Cedric Monfang, and who is in my thesis committee, Alexandra van Kemelen. And all this work are actually done by Chen, the postdoc in our lab, and also Alessia Santonza, who was PhD student and now in IRB Barcelona. And all the breast team members, and also my co-supervisor Orlando Sifrim and the 3 other bioinformaticians who are contributing to this project together by discussion and by giving advices. And we have a single cell and then the bulk RNA-seq facility and Institut Jules Bourdet, who were discussing about this project in the human manner. In the end, we didn't add it though, and pathologists who are always with us to understand the images. And thanks a lot again for the attending. And I would like to get the question if there is any question. Thank you.